recording.
Thank you, everybody who's joined us. It's really nice to have folk who hopefully will find this topic interesting. As Andy has said, I'm a senior clinical research fellow at the Patrick Wild Centre in Edinburgh, and I'm also a intellectual disability psychiatrist in the Edinburgh area. Today, I'm going to give you mainly a background to things that are relevant for genetic reports. So I'm going to start looking at the structure of DNA and how that links to genetic testing, and then Peter will follow on with more information about testing reports that you might get from clinical geneticists and talk you through some of the terminology with that. So overall, the aim is pretty simple, but hopefully we can make it accessible. So we want to try and explain this topic with minimal jargon. So I was just going to start with what is DNA. So there may well be people on the call who know quite a bit about this, but if you wouldn't mind just bearing with us, there may be people who would appreciate just going back to basics. So I think a lot of people have heard of DNA, and its full name is deoxyribonucleic acid, which you definitely don't need to remember. It is what's called a double-stranded molecule. So it has, don't actually know if you can see my pointer, hopefully you can. So it has these two strands, so there's one here on the left and one on the right, and they are joined by what we call nucleotide base pairs. So this outer bit is the backbone, and these are the nucleotide base pairs in the middle. And DNA can be found in the cell nucleus, which is like the hub of the cell, the control center, and also in tiny little parts of the cell called mitochondria, which have slightly different DNA, but I wasn't going to go into too much of the complexity of that just now. So you may also hear of chromosomes. So actually, if we go back to this previous slide, you can see that here on the right, with the coils of DNA and the colored nucleotides joining them, that's right down at the sort of really tiniest level of DNA. And then if you follow this up, it shows you how DNA gets coiled into different structures and then forms what we call chromosomes. So here in the middle, you'll see the word chromosome. So that kind of X-shaped blue shape is what a chromosome looks like if you look at it under a microscope. And that is useful to know because human cells have 46 chromosomes, 23 come from one parent and 23 from the other. So that is how you inherit genetic traits and characteristics. You may well have heard that chromosomes also determine whether somebody's biological sex is male or female. If they have an XY chromosome, which is what is shown here down on the bottom right, you see this what's called the sex chromosome here. And so for a male, it is XY, and for a female, it would be XX. All the other ones are grouped in size here, down from number 1 being the biggest, down through to 22 plus those sex chromosomes. And when you take that picture of all the chromosomes together, that's called a karyotype. So just a little bit more on DNA. So DNA is used like a blueprint in cells, and each part that codes from the DNA for a protein is almost like a recipe, if you want to think of it a bit in that way. And so that recipe or blueprint allows the DNA to instruct the cell to make proteins. And each part of the DNA that gives that instruction for a protein is a gene. And proteins are building blocks of cells, essentially. They have lots of different functions. They are made from long chains of smaller molecules called amino acids. And when they all get attached together, they sort of fold so that they're a particular shape, which means they can do whatever job that particular protein has been designed for. I also wanted to mention that each of these amino acids that is added to a chain when you're making a protein has a 3-letter code that denotes which one it is. Now, we're trying to keep this simple today, so there are things that we're not going to go into in huge amounts of detail, but it is important for later in my slides at least to just bear in mind that the DNA can be thought of in these sort of triplets, the way it codes for each amino acid. So when the cell is making the genetic, or using the genetic code to make proteins, it is reading it in these groups of 3, and they are called frames. Hopefully that makes enough sense. So I wanted to introduce you to the idea of DNA changes. I would imagine, although I'm not sure, that most people on this call are here because they are aware that changes in DNA or in genes can cause added difficulties for people. Obviously, our centre is particularly interested in conditions such as intellectual disability, Fragile X syndrome, and SYNGAP1, and these are genetic conditions. But I think it's important to note to start with that you can have changes in DNA which do not cause a problem. We all have differences in our DNA, or we would all be the same. There can be millions of changes of just one of these base pairs, these nucleotides I talked about before, that can cause no problems whatsoever. So that's just a point I wanted to make before we talk about the fact that there are other changes that can cause problems. So on this slide, you can see what we would say is just a sort of cartoon of DNA. And what I've done here is shown a single nucleotide variant. So you see here that the the green G has changed into sort of coral colored A. So that is a change in just one of these base pairs. And sometimes that can be enough to cause a problem. Other ones you might hear of are deletions. That's probably fairly self-evident, but it is when there are portions of DNA that are deleted. And that can be on a really small level, so you could even just have one base pair that's deleted, or you can have quite big chunks of DNA that are deleted. Perhaps the opposite of a deletion is you can get an insertion. So I've just shown here the point where DNA is inserted, which then leads to a longer strand and a change in the code. And again, that could well cause difficulties in the genes that we're interested in. That may mean things like intellectual disability, as I explained. You can also get a duplication. So you can have parts of a gene or parts of a DNA sequence that, for whatever reason, duplicate. So you get more than one of them. So you can see here that in this schematic, we now have 3 extra base pairs that duplicate the ones before them. You can get a situation where parts of the DNA split off and turn around, so that they're essentially backwards, and that's called an inversion. And on a chromosome level, so this is kind of on the ones I've spoken about already are down at the sort of tiny DNA level, but if you come up to the bigger chromosome level, which is where there's all the coils of DNA, you can have whole parts of chromosomes being deleted. You can also get what we call a translocation. And that is when a piece of a chromosome breaks off and attaches to a different chromosome. So you can see this here, that in this left-hand chromosome, this red part at the bottom, they can then break off and join on another one like it has here on the top right. And you can have that so that they are swapped around. So different pieces from one chromosome swapped with pieces from another. Now you might have heard of the word frame shift in terms of mutations. So this is where the fact that the DNA is read in these triplet 3 base pair frames is important. So I've tried to illustrate it here in as basic a way as I can. So if you look at the left-hand schematic, you can see that I've labeled 2 sets of brackets. So number 1 starts at the top, which means that if you're reading down the way, your code to start with is CAC, and then the second of the 3 is GAC. But on the panel on the right, I've tried to indicate what happens if you have a deletion at the start of this particular strand. And as you can see, that shifts the reading of the DNA down so that it now starts with ACG followed by ACT, which clearly is different from the example on the left. And it means that all the way through the DNA after that, everything is shifted. And as you can imagine, because it affects the whole DNA, there is really quite a significant chance that that could affect the protein or mean that there is no protein produced at all for that gene. One other thing I wanted to mention was called trinucleotide repeats. So there are some genetic conditions where what you see in terms of the change is that the DNA starts having a pattern of 3 base pairs that just repeat and repeat and repeat. Fragile X syndrome is one of these examples. And I've shown a little bit here of what that would look like, although in Fragile X itself, to have the really severe full mutation, you have to have 200 of these repeats. And obviously I did not have space for that. But what I'm showing is that you've here and on the strand on the right, if you start from the 3rd base pair down, you've got CGG. And then in a trinucleotide repeat, particularly of Fragile X in the CGG example, you just get CGG, 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 which obviously very much disrupts that gene and the protein it's coding for. So I'm going to mention some genetic testing techniques. Again, I'm going to do this in quite a broad brush way, but hopefully it gives you more of an idea of how it works. So I've already shown you a karyotype. So this is just a way of visualizing the different chromosomes. And because we know what they would typically look like, we can see if there are big differences in the different chromosomes. So a bit like those translocations where pieces could break off one and swap with another one. So a newer technique is called a microarray or a gene panel. And this is where you have DNA that is known as a reference sample. So DNA that doesn't have changes that we know to be harmful. So if you want to put it crudely, normal DNA, not that any DNA is normal in the sense that everybody's different, but this is in terms of looking for particular genetic conditions. So in this microarray, you have DNA from the patient and the reference sample that attach to fluorescent molecules, so molecules that will light up. And they then get put on a silicon microchip, which has many different DNA probes on it. And that means that it's looking for changes in particular parts of DNA that we know can be linked to, say, intellectual disability, or there are epilepsy panels or panels for other things. So it is looking at areas that we know are susceptible to changes that could be badly affecting somebody. And then the differences will be quantified by a computer. And now it is so sensitive that even if only one single base pair is affected by whatever the variant that person is carrying is, the DNA binds slightly less efficiently to the probe, and the computer is so sensitive it can detect even that small change in the fluorescent signal. Obviously, if you have a bigger change, then the signal difference will be higher. But now technology is so good that it can go right down to that single nucleotide level. The next thing is whole exome sequencing. Exomes are the parts of the DNA that, or parts of genes that code for proteins. So DNA is made up of the genes with the exomes, but these bits in between, these small lines that I've drawn here, are introns, and they do not code for proteins as such. They have other functions, but we know that the vast majority of variants or mutations, as you might call them, in human disease or human conditions come from the exons. So the testing is therefore done just on these exomes and not on all of the genome, all the DNA. And then you have the whole genome sequence, and that's where it tests the entire DNA. That is a lot more labor intensive, and it spits out lots of data that then takes a lot of time and money to analyze. So it doesn't tend to be the first step unless perhaps you've been in a research trial or something. So just whilst we're there, I'm just wondering if there's any burning questions. Andy, I don't know if there's anything in the Q&A. No, don't think so.
Not at the moment. I think you have explained it all so clearly, Lindsay. That's fine. But if people do have questions, they can just pop them in throughout the talk, and we can come to them at the end.
Great. Well, thank you. I will stop sharing my screen then and allow Pete to take over.
Right, thanks very much, Lindsay. So I will share my screen now. Get the right bit up. Yeah, so presentation in a second. There it is. Okay. So Lindsay's already done a lot of the work for me there. So I'm just going to do a little bit on genetic reports, and if any of you have received a genetic report, and my bit's about basically explaining a lot of the jargon that you might see in the report, and making it a bit clear what it might mean. Like Andy said, I'm working in clinical genetics. I'm a trainee, so I'm not yet at the sort of consultant level, but I've been doing it for a couple of years now. So hopefully I'm able to answer questions you might have about genetic reports and explain some of the bits that can be quite confusing about them. They can be very complicated, so even I find them difficult to understand sometimes for difficult conditions, and I work in genetics. So the one thing about these reports is that they're done at different labs in different parts of Scotland or Sometimes they're done in different parts of England, and the reports can look different depending on where they've been done. But they all have sort of some basic components to them, which I'll help break down. Some contain more information than others. I've done an example that basically should just highlight the important basic things that you would expect to see in a genetics report. So reports always have the identifying information. They should mention the reason for the test being done, what kind of genetic test was done. As Lindsay said, there's different types of genetic tests, and some tests only look for certain types of genetic changes. And the report will have a result, and it will have a conclusion as well. So just a little bit about the terms for changes in genes. Some of these terms can be used interchangeably by healthcare professionals, which can make things a little bit confusing. Variant, sometimes a change in a gene is called a variant. Sometimes it's just referred to as a gene change or a gene alteration. We generally don't use the word mutation anymore because some people might associate it with negative connotations. So we try to use variant, gene change, and gene alteration, but these all essentially would mean the same thing. So here I've got an example of a genetic report just with the patient identifiable information not present in it. It's quite small for me, but I think that's because I'm in presenter mode. But so I've broken up into different bits, and I'm going to explain what each of the bits mean. That's it, just zoomed in a little bit. So you've got a place where the patient information would be, the reason for the genetic test being done. In this case, it was seizures in a baby. Then it's what the sort of test being done is. So here it's an epilepsy gene panel. And then it's got a conclusion here, which summarizes the result. This is just the second half of the report zoomed in again. And I'll explain what each of these different bits mean. So, reason for the test, like I said, was seizures. And here the test done was a gene panel. And this is a genetic test that looks at a number of different genes that we know if you have a change in them, it can cause epilepsy. So we know this baby has had epilepsy, so sample of their DNA was taken, and they looked specifically at these 104 genes that we know are associated with epilepsy to see if there's any changes in those genes that look like they might be causing problems. And here's the result bit. And here the result is that they've found a change in this gene called SCN2A. Let me see if I can do a little point thing. Yeah, SCN2A, that's the name of the gene. And then there's this sort of confusing bit of writing here, which you don't necessarily need to understand, but I can explain if you're interested. So the C.1234 refers to the coding position on the gene. So that's, and I've just made this up so as not to provide any sort of single person's genetic results. So the C.1234 would be the 1234th letter in the DNA in that gene. And the A to G means that what would have normally been an A has been changed to a G. So this is like in Lindsay's example here. So this example, the G is changed to an A, but it's interchangeable. The letter is changed to a different letter, basically. And then the result of that is that Lindsay was mentioning amino acids. So the P here means that the result of this letter change in the DNA means the protein has got a different amino acid than what we would normally expect. So the amino acid is the building block, and the ileum, I think, is what ILE refers to at position 1, 2, 3 in the protein has been changed to valine, which means that the protein is got a different structure and therefore and is not working normally because of that. So, and this is the conclusion of that genetic report, stating that there's a heterozygous likely pathogenic sequence variant detected in the SCN2A gene. Now, what does this mean? So, heterozygous refers to the fact that we have 2 copies of most of our genes, and when there's a heterozygous gene variant, it means that in one of the 2 copies of that gene, there's a change in it that means it isn't working normally. And when it's this likely pathogenic bit, means that having this heterozygous change in this gene means that we think it's likely causing the condition that we're looking at. So, conclusion: this gene change in one of the 2 copies is likely causing the epilepsy that we've seen in this case. So I will go to the next slide. So you were noticing, you might have noticed I said likely pathogenic. And what that means is that it's very likely to be causing the condition in this case. Sometimes that, because as Lindsay was mentioning before, we all have different changes in our genes, and often those changes don't cause problems. Sometimes when we go looking in our genes for changes, we can find changes that we don't necessarily know if they're causing any problems or not. And we do lots of complicated science to try and work out if the gene change is causing the symptoms or not. And we end up coming out with a term to describe whether or not the gene change is causing those problems. So likely pathogenic means that it's very likely to be causing the condition. And we think more than 90% chance. But the person interpreting the report might, based on the symptoms, be more certain than 90%. So they might interpret the report with more certainty. And if it's pathogenic, that means that we're very confident that it's causing the condition. Sometimes we have variants that we're less sure about, called variants of uncertain significance, and they can be difficult to manage. But sometimes we find changes in genes that we don't think are causing any problems, and we wouldn't usually put them in a report because it's not causing any problems. We'll all have lots of changes, as Lindsay said, in our genes that aren't causing any problems. So, okay. And this is just the final line, kind of repeating what we said. Therefore, the sequence variant is likely to be the cause of the phenotype seen in this patient. Phenotype just means the symptoms that we're seeing in this patient. So the sequence variant is likely to be causing the symptoms in this patient. And just a bit saying they didn't find any gene changes in the other 104 genes that they looked at. So thank you very much. I hope that was clear, but I'm very happy to answer any questions that there are.
Thank you very much, Pete. There are indeed some questions that have come in. So the first one, and in part maybe this is coming from Lindsay's talk as well. So someone has asked, what's the difference between missense and nonsense mutations or variations? We see these things a lot on reports, so or people discussing them. I don't know if they're used from formally in genetic reports anymore, but families will often use those terms.
Shall I take that and you can correct me, Pete?
Sure, don't mind.
So a missense mutations, we're talking down at the sort of single base pair level, and a missense will be if there's been a change, and it's meaning that the protein isn't functioning as it should, perhaps because it's folded in a way that is not optimal. If you get a nonsense, then it tends to be more severe in the sense that it could stop the protein working altogether, or sometimes the protein won't be produced. Is that right, Pete?
Yeah, so the missense is like the one I described in my report, where it's changing the amino acid, so the protein might not work properly because of that, which is like what you said. And yeah, nonsense is where it's just what you described. Yeah.
And I guess a follow up to that is is one more serious than the other typically?
So we would. It doesn't necessarily mean that the outcome is more serious, or the symptoms are more serious. You might expect a nonsense gene change to be more likely to cause problems with the protein and therefore to cause the condition. But you can't really, I wouldn't be able to say with certainty that that's always the case. You might have a missense gene change that causes just as many symptoms as a nonsense change. It really depends on the gene and the protein and the part of the protein that the change is occurring in.
And then someone has asked about when someone's actually referred for NHS genetic testing, what do they actually get done in the first round? So someone with a kind of developmental condition, I guess, for this audience, what would be the kind of standard testing within the NHS for that?
So if it was for developmental delay, usually it would be the pediatricians that manage that, and they would often start with if they were deciding that they wanted to do a genetic test, which isn't always what they decide to do, but they would usually start with a microarray, which is the kind of test that Lindsay was describing, which looks for any big extra copies of DNA, like duplications, or missing pieces of DNA, like deletions. So that's usually what's done, and often there's also a Fragile X test done as well at the same time as that.
And then I guess a sort of a follow on to that would be when do people get selected for further kind of analysis? And someone has asked, is whole genome sequencing the only way to see every potential variation?
So it first one was when do we decide to do further tests?
Yeah. Yes.
So yeah, that's a difficult question to answer. It would depend on the picture that we're seeing and whether there is whether extra tests are likely to be helpful or not. The pediatricians might think that something is likely to have a genetic cause and therefore want to get more tests done, and that might involve a referral to clinical genetics, where someone like me or my colleagues would see the person and decide whether or not there's extra tests that might be needed, or the pediatricians might be satisfied from the test that they've done that they've excluded the likely genetic causes and that there's not likely to be any further use. They might also just write to us and ask us, let us know about it, and we might be able to advise them about that.
And this is a specific question someone has had about what does the C going to T mean in the C arrow T in the genetic reports? I think you had A to G. What C to T?
Oh, that's it's just another change that works in very similar way as the A to the G. So it means that the DNA sequence is changed, and that might mean that you get a different amino acid and a change in the protein as a result of the change in the DNA. And that could have very little consequences on the in terms of symptoms, or it could cause symptoms. It's difficult to say without doing all the science and like I was saying, and working out if the gene change is causing any problems.
Fine. And there was a question in the chat, I think, that came through where someone has asked about, it says, what about the reference? Is that important information in reports or not? Does that refer, I might be referring to the reference DNA sample, I guess, that's used for arrays. I'm not sure. Whoever's left that, they could put more information, maybe, if that's not what it is.
Sometimes, I don't know if this is what they mean, but sometimes when, oh, they just said never mind.
Never mind. Okay. I'm glad you knew what that NM meant. I was thinking, oh, what's that? Excellent. And then just going back, I suspect this one might be for Lindsay. It's specific to SYNGAP1, which is obviously one of the genes that we and she does a lot of work in here. We do a lot of work in but they said they know SYNGAP1 is at 6.21. Is there any further location data that describes and has an impact, i.e. more or less affected?
I suppose, so the 6 refers to chromosome 6, and then band 21 is a location on that chromosome. In terms of an individual's genetic change, so some of what Pete is describing is much more down at the granular level from that. So you would have the change within chromosome 6, band 21, because that's where SYNGAP is, but then the actual change within the gene would be down at that sort of DNA protein level he was describing. In terms of whether that has a link to the types of symptoms or problems or challenges that the individual has, there's a lot of people who are looking at this. I think we're starting to get a bit of an idea that different parts of the gene may link to more sort of clustering of things like language problems and level of intellectual disability, that kind of thing. There was actually a paper that was published last month that was looking at a sample of, I think, 44 people with SYNGAP. So that these samples are getting bigger and bigger, which does in time allow us to look more closely at the effect of a change in this part of the gene versus this part of the gene. But that's ongoing work and won't necessarily help any individual person. It certainly at the moment doesn't link to anything to do with, are there particular medicines that might help a particular person, or are there other things that we could do for a particular person on the basis of where the gene changes?
I think this question, I think you may just have answered this next question, which was about, is it possible to assign or match variants with severity of phenotype? So can you tell from a genetic change that's going to be more severe or less severe in terms of the phenotype of the clinical expression?
I suppose in terms of types of mutations, so if you have a frame shift mutation that throws the rest of the protein coding out of frame, then that's more likely to have a bigger effect. Whether you can necessarily predict exactly what an individual is going to look like from their genetic change, we don't have that level of detail. And as I say, we are starting, for example, in SYNGAP1, and that's the gene that I am most familiar with. It may be that in other conditions there's that perhaps are less rare, or there's been more research, that there may be a better understanding of whether particular changes in particular areas of the gene are linked to particular symptoms. As I say, that's very much evolving in SYNGAP at the moment.
Yeah, I think you know more about SYNGAP than me, Lindsay, definitely. But I would say that in some genes, in some conditions, there are particular variants that are known to cause symptoms more than others. But you can't always tell, even in those. Sometimes you get a gene change that can affect people very mildly, but in other people can have the exact same gene change and they're affected more severely. So it's really difficult to say.
Then this is a specific terminology one. What does monoallelic mean?
So we have 2 copies of most of our genes, but for some of those genes, even though we have 2 copies, one of the 2 copies is silenced. So only one of the copies is actually making protein. So that can mean we can have that just describes how the proteins are, how the genes are expressed, basically, and it can mean that we can have a gene change in one of the 2 copies, but because it's not the one that's expressed, then we don't have any symptoms from that. Or it can mean that we have a gene change in one of the copies, but that is the one that works normally isn't expressed normally. So that can mean symptoms are more severe. But it really depends on the gene and the sort of specific patient scenario. So it's hard to answer in broad terms what I'm not sure if you're referring to a particular condition there, Sarah, but I hope that's answered that a little bit.
Are specific genes typically monoallelic? Are they, certain genes which typically will, in most people where one will be switched off and, one copy will be switched off and things in there. Yeah.
Yeah, there are specific genes, yeah.
Okay. Fine. Okay. I think that's pretty much all of the questions that we have had come in. Let me just check across into the chat and things. No, yeah, I think that's pretty much all of the questions that we've had come in. There's one more. Sorry, one more.
I was going to say, hopefully that's because we've answered questions rather than either sending everybody to sleep or bamboozling you.
Creative questions. No, no. I mean, and if people still have, you can feel free still to answer or still to put them in at the moment. It says someone has written most of the Syngapians have a de novo variant. Is it feasible to see when did that change take place? I suppose it's worth just saying what a de novo variant is, I guess, in this context.
Yeah, so a de novo variant is one that is not a variant that hasn't been passed down from mom or dad. It's one that's occurred for the first time in the person we're seeing it in. The variant usually occurs just by chance, not because of anything anyone did. It occurs by chance when the sperm or the egg are formed, and then they go on to produce a baby, and that baby has the de novo variant. So you can't really. It's usually when the sperm or egg are formed, that's what a de novo variant is. I hope that answers that.
Oh, that's fine. That's good. Thank you. And I see that somebody's asked, just in relation, I think, to the discussion earlier on about, getting, if most people are tested by the microarray, but obviously a lot of the people with these sort of de novo or single base, changes, are only diagnosed if they manage to get exome sequencing or something in more detail. And someone's asked, how would a parent, how would a parent push a pediatrician for additional testing? Or I mean, another way of looking at that is, what are the kind of general criteria that might be used? And I know it's not necessarily fixed all the time, but what are the sort of criteria that are used to decide if someone might need to have more detailed or exome sequencing? And then I guess the next thing I'd ask is there going to be a point in time when exome sequencing becomes a standard, or genome sequencing becomes a standard within our own genetic services?
Yeah, so those are difficult questions to answer. I would say that pediatricians will certainly, if it's appropriate, seek for advice about extra genetic testing or refer to clinical genetics to have a discussion or to see if any further testing is needed. I know it can be very, very difficult if you are worried about a genetic condition and you've had testing done that hasn't found an answer. So I definitely sympathize with why you might want to have more tests done, but without knowing the sort of details of the case, it's hard to answer really whether genetic testing would be helpful. We've got a good service in Scotland where we can do genetic testing when it's needed, and extra genetic testing when it's needed. In terms of are we moving towards more whole exome testing? Not at the moment. I think because it's what we're doing right now is thought to be what is needed. So we don't want to do extra tests that aren't going to find extra, that aren't going to find anything new. In Scotland, we do exome testing, but we don't actually do whole genome testing in Scotland generally. And that's because, like Lindsay was saying, when we're looking at the exome, we're looking at all of the proteins, and the genome is the bits that aren't the proteins. So it's very rare that we'll look at the bits that aren't making proteins and find changes. So that could change in the future, but it's not happening yet. But if it does change, it's not likely to change to find many more problems.
Excellent. Okay. I think that is all of our questions. So first of all, I'd like to thank all of our guests for attending and listening and asking questions. I hope that has been really helpful to you. I know I have learned things tonight. I'd also like to thank Lindsay and Pete, of course, for coming and presenting and discussing these things so clearly. So thank you both. Thank you all for attending, and we'll end the event. I hope you enjoy the rest of your evening. Thank you.
Thank you. Goodbye, everyone.
Thank you. Bye.